name is Mariana Veloz, and I am a senior in the biotechnology shop, attending Bristol Plymouth High School in Taunton, Mass. Recently, we have conducted a lab called the PV92 Lab, and what the PV92 is it's an allo insertion, which is basically a genetic sequence in your DNA, and it truthfully doesn't mean anything. It just, some parts of it get passed down in ancestry, some just doesn't get passed down, and the whole goal of the lab is to determine if the students had this sequence in their DNA. So what we did was we took a hair follicle and we essentially broke up the hair follicle to release the DNA, and then we conducted PCR. And PCR is actually used in COVID testing, and what it does is it essentially makes copies of DNA, so that way if one copy messes up, you have another copy, and it makes hundreds and hundreds and hundreds of copies. So what we did was we took a copy of the PV92 and we ran it in agarose gel through gel electrophoresis. And what this process is basically a gel. It acts in a way as a molecular sieve, so that way any big chunks of DNA can't escape through. Nice thin small pieces of DNA can easily pass through the gel, and then you can be able to visualize and see what your results are. So what we do is we actually put in a standard in this gel, and that essentially says that the gel runs correctly, nothing bad happened to it. Next, you run our sample, and then we have to run our controls. And the point of the controls, it's extremely important. And what they do is they basically match up to see whether or not you have the PV92, whether or not you don't, whether or not one parent has it. And basically what I mean by one parent is when you receive your DNA, you get one from your mother and one from your father. So it can basically say that, one parent, one mother, or the father has it, and then the other parent does not have it. So what I actually ran it, and I came out as negative for the PV92, and I'll put the image up here and I'll explain that to you. So as you can see on the far left, there is the standard, and it seems to be the bands are nicely separated. Next comes to the DNA sample, which matches correctly with the homozygous negative control. And then you have the homozygous positive. And then on the far right, you have the heterozygous, which means both. So I hope you all learned something interesting today, and I'll be looking forward to hearing from you.